Welcome to our joint webinar featuring Pyxant Labs and Imabiotech. During this presentation, you will be hearing from two outstanding speakers, Mr. Shane Karnik, the Senior Laboratory Director at Pyxant Labs, and Dr. David Bonnel, Global Executive Director at Imabiotech. Before giving them the floor, each presentation will take approximately 15 minutes. You can ask your question in the Q&A box at the bottom of the screen, and we will dedicate time at the end of both presentations to answer them. This webinar is about leveraging the unique combination of bioanalysis and spatial bioanalysis to accelerate drug development. Mr. Karnik joined Pyxant Labs in 2003. As a senior laboratory director, his responsibility include leading a team of bioanalytical scientists conducting bioanalytical projects that support pharmaceutical drug development for both non-GLP and GLP preclinical and clinical studies. Dr. Bonnel joined Imabiotech in 2010 to install the platform and develop the analytical approaches. After being in charge of more than 300 studies during 10 years, he has been named the Global Executive Director of the Imabiotech Group. Pyxant Labs and Imabiotech offer robust regulatory development services and advanced innovative services. These global bioanalytical CROs combine Pyxant's best-in-class GLP and non-GLP bioanalytical services with Imabiotech spatial biology capabilities to create the first of its kind end-to-end solution throughout all the stages of drug research. Its unique services span from global bioanalytical to bioanalytical specialization and next-generation bioanalysis. And now the floor is yours, Mr. Karnik.
Great, thank you. Let me just queue up my slides here. Okay, and I'd like to thank Corinne for that introduction. And for putting together this webinar and inviting Pyxant Labs to be part of it. We're real excited to present some information on some case studies we've been working on for some unique problems. First, just
Shane, sorry to interrupt, David is speaking. I think you are sharing the one monitor. You have just to split.
Thank you.
Welcome.
Let me stop sharing one moment. Be 1 second. There's always something technical. How does that look, David?
Better.
Perfect.
Perfect. All right. Thank you. So just a little overview on Pyxant Labs. You know, Pyxant Labs has been around about 22 years. We're located in Colorado Springs, Colorado. We've just recently acquired a second site. It's in Salt Lake City. It's also a bioanalytical mass spec CRO site. It's a former Covance site, and prior to being acquired by Covance, they were Tandem Laboratories. Between our two sites, we have undergone 16 FDA audits. 7 of those have been conducted at Colorado Springs, and our most recent FDA audit was occurred this January 2021. It was for an NDA. That drug has been approved, and Pyxant Labs did not receive a 483 during that audit. So we're very proud of that. Between our two sites, we have a combined instrument platform of about 28 triple quad mass specs, 8 Q-exactive high-resolution mass specs, 10 UPLC fluorescence systems that we use for nucleotide analysis. But then obviously the traditional liquid handling systems and Watson LIMS database systems. Like I said earlier, Pyxant Labs is exclusively a bioanalytical CRO. We do everything from early stage discovery through phase 3 clinical trials and even post-market surveillance on drugs that are approved. As a lab goes, Pyxant Labs in a discovery setting does probably about 15% of our work is in tissue assays. So that's running everything from liver, any tissue you can really think of. I can't think of a tissue we have not run. We've run everything from cornea to bone marrow to, liver, kidney, all the normal nervous system tissues you could think of. We have a big focus on running oligonucleotides and regulated settings. We look at a lot of therapeutic peptides. We've done extensive work on antivirals. We look at a lot of lipid nanoparticle formulations for drugs that are being delivered. And then we also do a lot of protein analysis. So today I really want to cover 3 case studies where Pyxant Labs has used, mass spec to and get our clients some information they needed early in their drug discovery and development programs to be able to make some decision-making process. So the first one I want to talk about is a client that came to us with a need for an enzyme activity assessment. So they wanted to look at ornithine transcarbosylate enzyme and measure its activity level. But there was a need to be able to measure the enzyme's activity in a non-invasive measurement. So they wanted to use plasma in lieu of the typical tissue biopsy that is more traditionally done. There was also an understanding that with this work, there was going to be a lot of different samples from a lot of different subjects. And so there was a need to make sure that the endogenous level of the OTC enzymatic products was not going to bias the data, and we'd be able to compare data across subjects and also reproduce that data if needed for retesting. So a decision was made the OTC enzyme, and we run it in reverse, and we would drive it towards the formation of ornithine. So to do this, we use shrimp alkaline phosphatase to remove the carbonyl phosphate from the reaction to drive the reaction towards the formation of ornithine. So once we came up with the way we were going to measure ornithine, we still had the problem that there was going to be an endogenous level of ornithine, and it was going to be hard to tell what ornithine was created by the reaction and what was just endogenous to the system. So we decided to take an approach where we took citrulline and we had it labeled with deuteriums. We labeled it with 4 deuteriums. And we used this as a substrate to run the reaction in reverse. So we added citrulline D4, and then we monitored the production of ornithine D4, which by doing this, we were able to distinguish the difference between the ornithine produced by the enzymatic reaction and the ornithine that was natively in the system. And so after running this assessments, this enabled our client to get good enzymatic activity measurements so that they could show that the drug they were developing was actually targeting the enzyme of interest. That was very important to them. They didn't want to just understand if the drug was being just taken up and taken up, but they wanted to make sure that it was actually effective against the enzyme they were trying to elucidate a response against. The other thing to say is that after we developed this assay, we ran it in discovery for some time. Then we ended up validating it to be used in both GLP tox and for clinical use. The next thing I want to talk about are some work that Pyxant Labs has performed for liposomal nanoparticle formulations. So LPNs are used to encapsulate drugs to help deliver them to cells while minimizing toxicity. LPNs are used a lot in the oncology space for delivering chemotherapy drugs, but lately they've been in the news quite a bit because they're being used to deliver the mRNA vaccines. So if you think of Pfizer and Moderna's vaccines, they're using an LNP to deliver that vaccine to the patient. So some of the challenges when you're working with LNPs is really the client would like to know not only how much free drug was available in the sample, they'd also like to know how much drug has still been encapsulated. The other thing is that you cannot directly measure the encapsulated LNP containing the drug by mass spec. So there's some challenges there that you have to overcome. The other thing is these LNP molecules, they're made up of phospholipids, which are notorious to cause ion suppression and enhancements in mass spec instrumentation. So anybody that's been working in bioanalytical for some time knows there's quite a lot of work that goes into removing these phospholipids from a traditional small molecule assay because they've been known to, to cause enhancement or suppression of this signal over time. So what Pyxant Labs has come up with is when we get a sample and it needs to be measured for both the free drug and the encapsulated form of the drug, we will take solid phase extraction materials. We will selectively remove the encapsulated form of the drug from this sample. We will then capture the free drug. And then with these two fractions, we will take the one that contains the LPN. We will burst the lipid nanoparticle and thus getting the free drug. And that'll allow us to run the two samples independently. One will give us the total amount of free drug that was available in the sample. The other will give us the amount of encapsulated drug that was available. And then the client can use this data to better understand what their technology was doing in vivo. The other thing we can do is we can take these LPNs and we can actually measure the components that they're made of. So these are the phospholipids or cationic lipids, pegylated lipids that make up the LPNs. And these measurements are very important because the FDA would like to know where, how these LPNs are being eliminated from the body, and are they building up in any target organisms? So are they building up in the liver over time? And so by being able to run, plasma and tissue extracts for the lipid nanoparticle components, we're able to come up with assays that we have validated and used to support tox studies and clinical studies so that the client can show the FDA what is the fate of the LPN once it gets into the biological system. So for LPNs, we're really able to give our clients like a full understanding of what is occurring with their technology. We're able to give them a value for the free drug over time. We're able to give them a value for the encapsulated drug over time. But then we're also able to give them values for the lipid nanoparticles over time. So this allows them to look at the biodistribution of these materials, both in plasma and in tissue levels. The last thing I'd like to speak about today is some NRMA therapies that we have been working with in early development for some clients. So for mRNA therapies, the desire is to transcribe a human protein into a patient that is either underproducing the protein or is producing a form of the protein that is low in function. Early discovery work for this technology typically takes place in an animal model, and that animal model will have some form of native form of that protein that the researcher is looking to express. So these discovery programs will need to be able to distinguish between both the native form of the protein and the protein that's being transcribed. So typically when we talk about protein analysis, people immediately want to jump to some type of ligand binding assay. But in the case of early discovery work with these therapies, since there's going to be both the native form and the transcribed form of the protein, LBA sometimes is not selective or specific enough to tell the difference between the two. And sometimes the time required to produce the critical reagents is rate limiting in conducting the early feasibility studies. So we will take those studies and we will run the protein analysis by LCMS. So when you're analyzing proteins by mass spec, there's really two different options. One, you can try to measure the protein intact. There are challenges with this, the biggest challenge being the sensitivity that you achieve by measuring intact protein. It's usually pretty low and it's usually not going to allow you to assess the protein levels on, small individual sample sizes. So 20 microliters of plasma, it's going to be hard to run direct protein analysis and understand how much protein was in the sample. So instead, what is done is what's called a surrogate peptide approach. So we will take the protein, we will digest it, we will identify a unique surrogate peptide. We will take that peptide that comes from the digestion of the protein and we will analyze it. And then based on its values, we can go back and figure out the total amount of protein that was in the sample. So how is this done? So typically what Pyxant Labs will do is we'll get a recombinant form of the protein, we will digest it, and then we'll run it through our HRMS systems or OB-trap systems. And we'll run that in a data-dependent acquisition mode so that we can identify all the peptides that were produced after. We can also identify all the peptides and make sure they came from our protein of interest and they did not come from a protein that might have been a contamination of the recombinant form protein that got adulterated onto even the lab components that were used during sample extraction. So once we run these samples, this digested protein sample on our high-res mass spec system, we'll take that data and we'll run it through Skyline, and that'll help us identify the peptides that were created and trace those back to their protein of origin. It'll also allow us to identify which peptides are specific to the transcribed form or in the native form of the protein, so in the animal model. Once we get these peptides identified, we'll also run those through UniPort, and that's where we can identify that the peptide we're going to use for quantitation not only is unique to the protein we want to analyze, but it also does not occur in any of the proteome of the rodent or primate model that we're going to be working in. The reason for this is you do not want false positives because you're measuring a peptide that has come from a protein that is in the model organism. So how this data works is that we've digested the protein sample, we've then run a chromatography on it, and the software has identified all the peptides. The peptide identification is really an amino acid mapping where those peptides that were shown to be in high abundance, they're run through a collision cell where the Orbitrap can identify the Y and B ions formed, and that can go back and tell the exact structure and sequence of the amino acids that make up the peptide. So final thing I'd like to show here is just feasibility assessment we did for a client where they wanted to figure out if it was going to be possible to use LCMS to distinguish between a human form of a protein and a primate form of a protein, because they were going to be working in a primate model. For this work, it was really important that we were able to show not only that the human protein was present, but also that the monkey protein was present. So for proof of concept, what we wanted to do was be able to measure both the native and the transcribed protein in a single plasma sample. So to do this, we took plasma from human and monkey, and we mixed those in increasing ratios. So we started with a small ratio of human plasma, added that to monkey protein, and increased that till we got to about 75% human plasma and monkey plasma. We then took these samples, we digested them, and then we monitored them for two different peptides. One peptide that was unique to the native form of the protein, another that was unique to the transcribed protein. So I show the kind of a redacted amino acid sequence, and you can see that the two peptides that we followed differed by one amino acid. So when we ran this data through our mass spec, and at this point, we're no longer using the high res, we've moved this over to triple quad mass spec. You can see, as expected, as the ratio of human plasma increases, we also see an increase in the amount of the human form of the protein. And conversely, as the human plasma concentration goes up, the primate form of the protein decreases. So with this data, we were able to show that LC-MS was selected for the 2 types of peptides and could be used for early development. This also allowed our clients to begin development without developing an LBA assay and investing time and money into producing the critical reagents for that type of assay. So that brings me to the end of my talk. So I'd like to thank everybody for listening today, and I'm excited to hear what David has to present. So I will.
Thank you, Mr. Karnik. As I mentioned at the beginning of your presentation, if you have some questions for Mr. Karnik, you can ask those in the Q&A box at the bottom of the screen, and we will answer the question at the end of Dr. Bonnel's presentation. So Dr. Bonnel, the floor is yours.
Many thanks, Corinne. Many thanks, Shane. Let me share my screen. Yeah. Can you see my screen, Corinne?
Yes.
Perfect. Thank you very much. Thank you, Shane, for this, I would say, nice example in bioanalysis. And what I will try to do now is to jump in the spatial bioanalysis part of our services in the group Pyxant plus Imabiotech, and you will see the complementarity between our different approaches, and you will understand why we have this unique, I would say, set of services right now in our global zero. So first of all, just to let you know that when we are speaking about spatial bioanalysis, we are speaking about solid tissues. So we will directly work on tissue sections. And as you can see on this slide, we are able to work on isolated tissue section from preclinical studies, of course, from animal models. So you have some example here of isolated organs. We can also work directly on whole body, rat or mouse if needed, and I will show you some example. And from these preclinical tissues, we will be able to extract, as you will see, some information in terms of molecular distribution, etc. And we will be able to jump with you on the clinical studies if we have access to the biopsies in order to use our platform to follow directly some molecules of interest in the tissues. So we are routinely working, for example, on core needle biopsies in oncology, for example, in order to see where is localized a compound, what is the effect of the compound on some specific markers. I will enter a little bit more in the details in the next slides. So as illustrated before, we can work, of course, on fluids. We can work on tissue homogenate with the Pyxant services, and we can work on solid tissues with the Imabiotech services. In terms of spatial bioanalysis, you can see on this slide, I would say, an overview of the types of molecules that we can detect with our platform. So, of course, we will work on drug and the related metabolites of the drug directly in the tissue section. A quick note to mention that we can also work on elements and follow directly the distribution of elements again in the tissue section. And in addition to the drug, we will be able to monitor directly some biomarkers, again, on the solid tissues by using the right technology. So we can do directly some transcriptomics, proteomics, lipidomics, metabolomics analysis directly on tissues. So for this, we will use different types of instruments, and we will select the right instrument depending on the need. We can, of course, at the end, combine the results from the different platforms in order, for example, to combine the drug distribution with a target distribution and confirm, for example, a target exposure. So how it works, we will always start from a solid tissue, and we will also always start from a tissue section. The objective is to preserve the tissue microenvironment, to preserve the histological information. And from this tissue section, as illustrated here with the tumor tissue section, we can select the right platform depending on the molecules that we have to target: elemental, molecular imaging, RNA, protein profiling, or the imaging mass cytometry, if we want to target some cell types or some specific markers. We are also able, of course, to perform any type of staining, immunostaining, I would say classical microscopic approaches. And if for any reason we cannot use the imaging platforms, we have the possibility to micro dissect some regions of interest directly on the tissue sections. The idea is the same. Be sure that we preserve the localization information. And on this micro dissected region, we can, of course, combine our effort with the bioanalysis parts in order to quantify the amount of the molecule of interest in each microdissected region, or we can also perform some gene expression profiling directly on these isolated structures. So let's enter a little bit into the details of each platform, and I will try to give you some examples. So first platform that I would like to show you, this is a laser microdissection, LMD. So as I mentioned before, the idea here is to know where we are in the tissue and be able to isolate, as illustrated here, the regions of interest and to couple this with the LC-MS as illustrated before by Shane. So by doing this, we can exactly quantify the molecule of interest in the region of interest. In addition to this, we can also target with this unique platform the gene expression to monitor, for example, the impact of a treatment on some gene expression or to compare different, I would say, conditions. The first example for this, it's a work that we have done directly on some CRC tumors. The objective here was to visualize and to detect, for example, the level of expression of some specific genes between an early stage of development of the tumor and a late stage of development. So we have microdissected the tumor cells directly on tissue section from both conditions. We perform an RNA extraction, an amplification, and a DNA quantification. And at the end, we have targeted a specific panel of genes, pan-cancer progression panels, in order to see the differences in terms of gene expression between the early stage and the late stage. So here you have an example of the results that we have obtained directly on this study, with the possibility to very quickly identify some pathways based on the gene expression, pathways of interest in oncology, of course, in this case, and to visualize the upregulation or the downregulation of these genes in this specific late and early stages of development. And here on this volcano plot, you can see that we have also access, of course, to the markers, and we can see which markers is upregulated or downregulated. It is super useful, again, if you want to characterize a specific disease or if you want to confirm the efficacy of a compound based on the gene expression. The next platform that we are using, this is a mass spectrometry imaging, MSI, and this approach is quantitative. So how it works, we will directly work again on the solid tissues. We will perform some tissue section. So for example, here on a mouse cell body or on a kidney on this picture. And we will put this section on a slide. We just spray the matrix onto the top of the tissue. And then the slide with the tissue section is introduced into a mass spectrometer, and a laser will shoot in point-by-point mode of acquisition. And for each impact of the laser, we will obtain one spectrum. And in this spectrum, you have all the molecules detected at the exact laser shoot position. Based on this huge data set, with all the molecules detected, we have then to target the M over Z, the ion, the molecule of interest, to generate automatically the biodistribution of this molecule on the tissue section. So for each molecule that has been detected, we can generate the image of this molecule as illustrated here on the mouse whole body. So the main advantage is really the specificity. There is no radioactivity, no fluorescence. We can directly target the drug, for example, of interest. This is quantitative. We can quantify the exact amount of drug, for example, in a region of interest. And the best spatial resolution that we can achieve is at 20 micrometers with this approach. One limitation, this platform is really dedicated, I would say, to small molecules, drug, lipids, metabolites, peptides. It can be challenging to detect some proteins. We have this expertise as well, but it can be challenging depending on the target that you do have. So how we are using this in spatial bioanalysis? An example is the whole body PK approach, whole body pharmacokinetic. We have a lot of projects ongoing for which we need to visualize the biodistribution of a candidate or a lead or a molecule in development directly in the whole body at some early stage or late stage of development. So to do this, we are directly working on the carcasses of the animals, and we have also access to the plasma, of course, of the animals. And we will combine the QMSI with the LC-MS approach. The QMSI approach will be used to visualize, for example, the drug distribution in a specific section plane of interest at different time points, for example. So we have access to the kinetic. And we can directly visualize the distribution of the compound in the mouse whole body section in each organ over the time. If needed, and if possible, we can also detect some related metabolites of the molecule due to the specificity of the approach. So for example, in this example, you have at T2 hour a nice accumulation of the compound in the brain, and you can see that the main metabolite is not distributed in the brain of the animal. And you can follow the distribution, the kinetic of distribution over the time. And by combining this result with the plasma information, we are able to generate the PK profiles of the compound in each organ. And we have access, of course, to all the PK parameters of your molecule directly in all the organs. So it is really useful if you want to have access to this information in order to anticipate some potential safety concern. For example, with some unexpected accumulation, you can also confirm the potential efficacy to visualize the expected accumulation in a specific organ of interest. You can have some xenograph tissue as well on the whole bodies. So it is really used at some early stage to anticipate and to confirm the efficacy. You can compare different candidates, or you can also evaluate different routes of administration, for example. A quick note to mention that with these platforms, the quantitative mass spec imaging, we can of course target exogenous molecules as illustrated, but we can also detect endogenous markers. And we are doing this in several years right now at Imabiotech. And by having this expertise, we can directly target some metabolites, lipids, amino acids, neurotransmitters of interest directly in the tissue sections. And we can see what is the distribution of these molecules. And we can, for example, see where is the compound, where is the drug, and if there is a decrease or an increase of some specific metabolites in the tissues. So it allows really to do some pharmacodynamics analysis directly on the tissue section in preclinical and in clinical studies. The next platform that we are using is more dedicated to the proteins. So here we will speak about large molecules. So this is the imaging mass cytometry. By using this technology, we can go up to 40 different protein markers directly on one tissue section again. The objective is to detect the different proteins, the different markers, with some antibodies that will, of course, target these markers, and we will put a metal tag onto the top of each antibody. And by using the ICP-MS instrument, the Hyperion system from Freedyne, we will directly detect the metal tags, and the distribution of the metal tags will reflect the distribution of the markers of interest. The main advantage is the fact that you have really a multiplex approach. You can detect up to 40 proteins simultaneously, not just 1, 2, but 40, and you can definitively characterize your sample and see if you have some changes or some evolution of your specific cells infiltration, if you have some specific modulation of pathway directly in the tissues. So here you have a non-exhaustive list of cells that we can target. And in terms of illustration of this, example of this with the IMC, here you have an example of a cells composition analysis in some diabetes tissues. So by using the IMC approach and by targeting 35 markers directly in the tissues and by focusing on the highlight of the pancreas, it is possible to directly, for example, characterize the composition in terms of cells directly in the highlight from a control, I would say, group without any diabetic, with an early stage group of the disease and a long-term stage of the disease. And you can see the destruction, I would say, of the islets over the time. And if you look at the result here, you can see that the beta cells are destroyed over the time thanks to the disease progression directly in the samples. And by having access to this information, we can characterize the cell's composition. In addition to this, we can directly target, based on the other markers targeted with these panels, we can target, for example, the immune cells, and we can, I would say, quantify the amount of immune cells based on the cell density per millimeter squared. And we can see, for example, the recruitment, let's say, of the T cells directly at the early stage of development of the disease. Same mechanism for the macrophage. What we can also do is to calculate the distance between the immune cells and the islets to see the progression of the infiltration from the immune cells to the islets of the pancreas. And here you have a nice example with the distance of each immune cells in the different groups. And you can see here that the cells are, I would say, really close to the islets in the disease group for the immune T cells compared to the control group, for example. So very quickly, we can characterize the composition of a disease. And of course, if we apply a treatment on top of this, we should see the modulation of the infiltration of the cells. We can characterize the, I would say, efficacy of the compound. The next platform that I would like to show you, this is a multiplex in situ profiling platform that we are using at Imabiotech, the GeoMx system from NanoString. So this platform is a little bit similar to the previous one. You can go up to 80 markers simultaneously. We are able to customize the panels of genes, I mean RNA or proteins that we can target directly. Technically, there is a difference. We will use 4 fluorescent antibodies to target 4 markers, of interest. And then based on these images, we will go in a specific region of interest based on the 4 fluorescent antibodies used. And in this region of interest, we will extract the let's say, flag coming from antibodies or oligoprobes that have been incubated on the tissue section. And by extracting, I would say, the flag, the oligo flag, we will collect and dispense this flag into the system to measure the amount of marker per histological region of interest. So you don't have an image at the end, but you have some heat maps or any statistical, I would say, illustration that will reflect the abundance, the level of expression of the proteins or the RNA per histological region selected on the tissue section. So an illustration of this is a combination of mass spec imaging, the previous platform, H&E staining, and the GeoMx system directly on tissues, on tumor tissues. And by doing this, for example, here, by using the QMSI to target the lactate and the pyruvate, the lactate is a modulator of the immune, of some immune chain points. So it can be of interest to see where is accumulated the lactate, for example, in a tissue, in a tumor tissue section. And by targeting this by MSI, we can directly select some regions of interest, for example, low pyruvate, high lactate level, or the opposite. And we can specifically look at the, I would say, expression of some markers or protein markers in this region with a high level of lactate or low level of lactate, for example, and by using the GeoMx system. So it is what we have done here on a specific panels of interest with some specific cells or markers. And by doing this, we have been able to identify, for example, some hot phenotype related to the disease when you have an accumulation of the lactate, or some cold phenotype when you don't have an accumulation of the lactate. So by combining this information, you can definitively see, for example, in this case, the modulation of the T cells, if they are exhausted or non-exhausted in the different regions. So if your compound is playing a role on the lactate pathway, we can see the efficacy of the compound directly on the pathways. So it was my last slide. To conclude, I would say to conclude the general presentation, what we are able to do today at Pyxant and Imabiotech is really to combine bioanalysis and spatial bioanalysis. So we have the unique expertise to target some drugs, oligo, peptide, proteins, small molecules, etc., directly in fluids, in solid tissues, to give you access to the, I would say, the peak information or the bioavailability, excuse me, the biodistribution, any peak information that you can need from preclinical to clinical stages. We can confirm the target exposure. We are working in GLP tox, of course, projects. And, excuse me, at Pyxant, we are working a lot on clinical samples in order to follow directly your molecule of interest in some GLP, in a GLP environment. In terms of biomarker, we are able, as you have seen in our presentations, to follow some biomarker directly in discovery, to do some pharmacodynamics analysis. We can study the mechanism of action. We can analyze the tissue molecular heterogeneity, the microenvironment phenotyping, or the pathway modulation directly in the tissue microenvironment. And again, by combining both the bioanalysis and the spatial bioanalysis, we can see where is the compound and what is the effect of the compound directly in tissues. So I really would like to thank you for your attention, and I think Shane and myself, we are available if you have any question. Corinne will let you ask us a question. Many thanks.
Thank you very much, Dr. Bonnel. Yes, we're gonna take some question. The first question is for you, Mr. Karnik. Someone wants to know what is the duration of LNP project, including the development as well as the validation. You're mute, Mr. Karnik, sorry.
Oh, perfect. Sorry about that. Yeah, so duration. So for development, we're typically looking, obviously depends on the complexity of the drug, the complexity of the system, but 10 days would 10 laboratory days to do development. Validation is typically is going to be another 2 weeks after that. Then obviously in between there's some reporting and other timelines that need to occur. So for a, how we always call it, from the signing and receiving of material to you getting a validated report would be 6 to 8 weeks, a validation report, just with the things that have to occur there, like QA review of that report, drafting of the report, getting comments back from the client. But if you're looking for a quick discovery work, we can typically turn that around in just in a few weeks. Is that helpful?
It's very helpful. Thank you. Dr. Bonnel, for the IMC technology, is tissue fresh or paraffin embedded?
Good question. Yeah, I didn't enter into the details. So we can work on both. It will really depend on the panels of antibodies that we have to design, or of the antibodies that are available. So we can work on both, but again, the markers that you will target can maybe help us to select if we prefer to work on FFP or on frozen tissues. A quick note, for each platform that I have introduced, we can work on both types of, I would say, tissues, fixed and paraffin embedded and frozen. Only for the mass spec imaging platform, we have clearly a preference for frozen tissues because the fixation step can lead to some delocalization of molecules, small molecules. So this is the only platform for which we do prefer to work on frozen tissues.
Thank you very much. Mr. Karnik, what are the validation criteria?
Yeah, so with most of everything I described there, so when we talked about the OTC, enzyme activity assessment, we followed the FDA bioanalytical method validation guideline, so the 2018. So, you're going to look at accuracy, precision, selectivity, specificity, recovery, and all the stability experiments that go with that matrix effect additionally. The thing, we looked at when we did the OTC assay. So when it's something that's a little more unique, we're going to do a full validation, but there are going to be times unique experiments need to be made. So in that case, we actually tested incurred samples of plasma, and we characterized those, some that had low enzymatic activity, some that had high enzymatic activity. And then we used those as run controls throughout the validation. So even though we were running accuracy precision, we ran the same incurred samples with each validation run so that we could show that the method was reproducible over the 3 analytical AP runs. And then we kept those same incurred QCs, if you will, and we use those all the way through sample analysis. And again, whenever you're doing a validation of these types, you're going to want to stay as close as you can to the FDA validation or the EMA guidances. But obviously with these kind of unique things where you're measuring like enzymatic activity, or you're measuring, the transcription of a protein, there's going to be some modifications made to those. So that's the one thing I think Pyxant Labs does pretty well. We have scientists, our typical senior scientist has 21 years in regulated bioanalysis. We've kind of seen every type of situation that's occurred, and when we need to adjust the validation protocols for a unique type of analysis, we can come up with the good experiments that are going to show the regulators that, the method really was fit and it is going to give good, reliable data. So,
thank you.
Obviously, intent guidance that's coming out, so that's going to change. We're going forward in the future too.
Thank you very much, Mr. Karnik. Dr. Bonnel, a question for you. Can you confirm a target engagement?
We are speaking about a target exposure, not a target engagement.
Sorry.
So the question is nice. I mean, the target engagement is really when you can show that you have an interaction between both molecules, the drug and the target. We speak about target exposure at Imabiotech because we can colocalize, for example, the drug by mass spec imaging, for example, and the target, a receptor, an enzyme, any type of molecule, for example, by immunohistochemistry. And by having an overlay between both the drug and the target, we can confirm that the drug is able to reach the target region, and we can quantify the coverage and the amount of drug colocalized with the target. We are speaking about a target engagement if we can follow some readout markers. For example, if we can see this colocalization, and if we can see that there is an effect of the drug, an inhibition, an activation on a pathway. In this case, we normally do know that the drug is active, is playing a role on the target, and we can follow this by, I would say, analyzing some metabolites, lipids, proteins, genes, cell types, as you have seen. So target exposure, if we are not able to see the readout markers, target engagement, if really we can do it.
Thank you very much. And the last question is going to go for you, Dr. Bonnel. What is the advantage of the IM cytometry approach compared to, for example, detecting the metal tags with tough SIMS imaging?
Ooh. Good question. I would say maybe the robustness of the system. So that's a good question, a good technical question. The advantage is really the system itself. So the robustness, the throughput, etc., and the fact that you have some panels available, etc., etc. So you can, of course, with some tough SIMS approach theoretic targets, metal tags, and see their distribution. Now, the most important is to have the software in order to do the data analysis and be able to extract the information. As you have seen in my presentation, that's a very important part. So I would say the instrument is important, of course, so the throughput, the robustness, and then the data analysis and how you will interpret, extract this information, and some software dedicated to this on the imaging site of systems that we are using at Imabiotech.
So this is going to end our webinar session today. So I would like to thank everyone for joining us today, and thanks the two outstanding speakers. If you need to ask some additional question, you can see their email address directly on screen. And hopefully you will receive, like very quickly a new email to join a new webinar. So thank you very much for joining us today. Thank you.
Many thanks. Bye-bye.